Until recently, researchers assumed that communication between distant tissues involved either neural impulses or secreted hormones distributed via the blood. However, another communication pathway has been identified, which allows for the exchange of transmissible macromolecules between cells via an extracellular vesicle called an exosome. These exosomes have been associated with the exacerbation of various neurological diseases by promoting the spread of misfolded protein aggregates to aggregate-free cells. In essence, exosomes play a role in promoting the conversion of healthy cells to pathogenic ones.
Exosomes are small membrane vesicles within a larger vesicle known as a multivesicular endosome, or MVE. To understand how exosomes form, focus on number 1 in the figure. Now, we've already learned a lot about extracellular vesicles that bud from the plasma membrane. However, exosomes form when an MVE buds inward and captures bits of cytoplasm, including free-floating proteins, mRNA, and miRNA, forming small internal vesicles.
Originally, it was thought that the only role of exosomes was to help traffic extracellular molecules to lysosomes. Number 3 shows how an MVE fuses with the membrane of a lysosome, which then degrades the exosomes and its contents. However, in the mid-1980s, researchers discovered that MVEs also fuse with the cell membrane, releasing exosomes outside the cell as shown by number 2. Exosomes can then travel to distant target cells where they bind to specific ligands on the cell membrane. While exosomes are secreted by all cell types and assist with positive actions such as tissue repair and neural communication, exosomes containing aggregation-prone proteins have been found in the blood and cerebrospinal fluid of patients affected with neurodegenerative disorders. Deposits of misfolded aggregated proteins is a common feature of neurodegenerative diseases. Evidence suggests that exosomes act as a protected environment that promotes the aggregation of misfolded or pathogenic proteins such as prion and amyloid deposits, and allows the distribution of these protein aggregates between cells.
One such class of diseases in which this occurs is prion diseases. Prion diseases, such as Creutzfeldt-Jakob disease, or CJD, a neurodegenerative disease characterized by rapidly progressing dementia, vision loss, and cerebellar ataxia, occur primarily through the aggregation of misfolded prion proteins into a disease-transmissible form. Research has indicated the presence of these misfolded proteins aggregates in unrelated cell types, which signifies the possibility that exosomes allow for the transmission of misfolded proteins from the peripheries to the central nervous system. PRPC is a prion protein that, when misfolded into PRPSC, plays a large role in CJD. A GPI linker protein connects each prion protein to lipid rafts in the membrane. PRPC is taken into the cell via endocytosis and is contained within an early endosome. It travels in the endosome and fuses to a large multivesicular body with the help of ceramide and escort proteins. Normally, PRPC is sent to the lysosome for degradation, but in Creutzfeldt-Jakob disease, the multivesicular body releases exosomes containing PRPSC back into the extracellular environment.
Another example of how exosomes play a role in the pathogenesis of a neurodegenerative disease is seen in Alzheimer's patients. Extracellular amyloid precursor protein, or APP, contributes to the generation of plaques in the brain, which are characteristic of Alzheimer's disease. Amyloids are extracellular protein aggregates that are insoluble and resistant to degradation.
APP is cleaved by beta secretase on early endosomes, which forms secreted amyloid precursor protein, SAPP beta. From there, gamma secretase cleaves SAPP beta into amyloid beta, or AB. Amyloid beta is responsible for forming these aggregates, which are beta sheets that are self-assembled and propagated throughout the cell. From there, amyloid beta is trafficked into multivesicular bodies, and the cargo is released from the cytoplasm into the extracellular space.
Now, researchers are working to answer 3 important questions. Through what mechanisms are aggregates packed into exosomes? Considering the high concentration of lipids in exosomes, what is their role in protein misfolding? And finally, how are exosomes taken up by recipient cells? To answer these questions will fill crucial gaps in current literature and increase our understanding of the role of exosomes in neurodegenerative diseases.